cytoplasm remains connected between the adjacent cells. Helps in maintaining protoplasmic continuity with adjacent living cells. Function. Concerned with transport of material and transmission of stimuli from cell to cell. In parasitic plants like Cuscuta, Loranthus and so on Plasmodesmata, connections exist between their hostoria and host cell, enabling them collecting nutrients. While well developed, Plasmodesmata are seen in endosperm of some seeds like Phoenix. And we will upload videos on them certainly. Importance of palynology in modern classification taxonomy. The information generated by palynology is highly useful in arranging several disputed plant genera, as well as species. Pollen grains of fossil plants are also used to solve problems in plant taxonomy. Basic features of pollen grains including morphology along with the ornamentation of pollen exene is useful in classifying and identifying plants. Note: among the groups angiosperms are having the largest variation in pollen morphology. Pollen analysis data could be used for identification and classification of plants. Microspores are one of the conservative of plant structures for taxonomical studies. Stenopolinus and Europolinus are the two family categories based on pollen type. Stenopolinus, characteristic and constant pollen, that is single morphological pollen type throughout the family. Example: Cruciferae. All members have tricolpate and reticulate pollen grains. Europolinus have significant variation in pollen morphology among the different genera of the family. Example: Euphorbiaceae, Phytolaccaceae, Betulaceae and Liliaceae. Note: pollen analysis, NPC analysis are increasingly used for effectively solving problems of taxonomy as well as for the identification of new species. There are also applications of pollen study like the pollen analysis in forensic science. Branches. Primary growth. The increase in diameter of axis as a result of the activities of lateral meristem. Secondary growth. In dicot stem. Secondary thickening. Vascular bundles collateral, open and limited in number and arranged in the form of broken ring also possess the cambium, which is the main reason behind the secondary growth. The stellar secondary growth sooner or later followed by the extrastellar secondary growth completes the process of normal secondary growth. Stellar secondary growth. The cambium which lies in between xylem and phloem in each vascular bundle called fascicular cambium is the reason behind secondary growth in steel and the tissue formation in steel. Stellar secondary growth. Steps. A. Medullary ray cells mostly in line with fascicular cambium becomes meristematic and forms the interfascicular cambium in between two vascular bundles. B. Interfascicular cambium joins with intrafascicular fascicular cambium to form complete ring, cambial ring, vascular cambium. Note: the vascular cambium consists of two types of cells, fusiform initials and ray initials. C. The fusiform intials of the vascular cambium undergo successive tangential divisions and produce xylem mother cells internally and phloem mother cells externally. D. Thexylem mother cells by division and differentiation develops to secondary xylem and phloem mother cells to secondary phloem. Note: usually cambium produces more xylem than phloem. E. The formation of secondary xylem and phloem creates much stress in interior of stem. Consequently, the pith may be deformed and obliterated by inward pressure of the secondary body. F. The ray initials give rise to xylem and phloem rays, vascular rays, which helps in the movement of water from xylem to cambium and phloem, transportation of food from phloem to cambium and xylem, and also makes gaseous exchange with Outer atmosphere through intercellular space is easy. Secondary xylem, as like primary, helps in conduction of water and mineral elements and provides mechanical support. Xylem parenchyma helps in storage. Note in old trees tylose is formed inside secondary xylem vessels and tracheids. Makes them function only for mechanical support. Secondary phloem. Phloem got crushed during secondary thickening. The secondary phloem takes over the function of conduction of food and remains active for a considerable period. Note, unlike xylem, phloem does not have a distinction of early and late. Phloem. Extrastellar secondary growth. Outside steel steps. A. Epidermis gets stretched and ruptured due to the stellar pressure on cortex and epidermis. Sometimes it breaks down altogether. B. To replace epidermis, new protective tissue periderm is formed by activity of meristematic tissue, phellogen cork. Cambium. C. Phellogen produces more cells to outside phellum or cork protective in function and less cells to inside. Parenchymates living phelloderner. Secondary cortex carry out photosynthesis and storage of food. Note phellogen, phellum and phelloderm together constitutes the periderm. D. Bark when sometimes the pressure from steel becomes inadequate. Additional layer of periderm forms in the deeper regions of cortex, pericycle, phloem etc. and results in the formation of bark. E. Lenticels. Some pores raised. Develops on surface of stem for exchange of gases. When epidermis is replaced by periderm. Here is the diagrammatic representation of stellar and extrastellar secondary growth in dicot stems. Secondary thickening in monocot stems. Monocot stems and roots generally do not undergo secondary growth due to the absence of cambium. Secondary growth in monocots. Monocots generally do not undergo secondary thickening because of the closed vascular bundles due to absence of cambium. In some families like Liliaceae, Amaryllidaceae secondary growth, anomalous in origin and function occurs. This secondary growth is the result of the formation of special meristematic tissue called secondary cambium which develops from the inner cortical as a result of the activities of lateral meristem. Secondary growth. The processes of secondary growth that give rise to this type of structure may be termed the common type of secondary growth. However, in many P1 ants there are deviations from this type of secondary growth. Such deviations may include such features as a. The unequal activity of different portions of the cambium on the circumference of the axis. b. The alteration of the relative amounts and c. Position of the xylem and phloem, and d. The appearance of additional cambia. The secondary growth that results in the development of a secondary body differing from the common is termed anomalous secondary growth. Here you can correctly view the difference between secondary as well as the anomalous secondary growth in this slide. Anomalous secondary growth in dicot stem. A. Bignonia stem. Bignonia is a dicot woody climber belongs to family Bignoniaceae. Anomalous secondary growth in this stem is due to abnormal behavior of the cambium during later stages. Secondary growth in Bignonia stem is not typical of that found in Dicotyledonous stem. In some plants like Bignonia and other members of the family Bignoniaceae, the cambium is normal in position and activity for the first time, but immediately it cuts off different proportions of xylem and phloem at 4 alternative points in form of a cross. The cambium produces secondary xylem and secondary phloem in variable quantities, in some segments much greater amount of xylem than of phloem and in others more phloem than xylem. As a result, a characteristic structure is formed with ridged and furrowed xylem cylinder. The ridges are wider than the grooves. In the secondary phloem formed in the grooves, parallel strips of bast fibers are formed. The cambium breaks up into a number of strips. The wider ones remain on top of the ridges and narrower ones remain at the base of the grooves. The number of wedges formed is usually 4 in number, symmetrically arranged corresponding in position to the larger primary vascular bundle. This is a kind of adaptive anomaly exists in Bignonia because of its habitat that is of a woody climber. Now it's time to view the detailed view of TS transverse section of Bignonia stem showing the specific anomality. And here is the CS cross section also. Hope this helps. B. Boerhaavia stem. The abnormal secondary growth from normal cambium. In this type, cambium of normal type is present and persist but by peculiarity or irregularity in its activity develop vascular tissues of unusual arrangement. In the vascular bundles of outer ring, Strips of cambia are formed between the vascular bundles, which fuse with the strips of the fascicular cambium and thus form a continuous ring of cambium. The fascicular cambium produces secondary xylem to the inner side and secondary phloem to the outer side. The interfascicular cambium give rise to the conjunctive parenchyma to the inner side and secondary parenchyma to the outer side. Very soon the activity of this cambial ring stops. Later a cambial ring develops secondarily from the pericycle and becomes active. It cuts secondary phloem towards outer side and secondary xylem towards inner side. In between them conjunctive tissue is produced. As a result, a ring of collateral bundles separated by conjunctive tissue is formed. After a period of activity, this cambium also stops to function. Another ring of cambium arises outside, which behaves in the same pattern. Thus, in this stem several rings of cambia arise successively in a centrifugal manner. The abnormal cambial ring produces xylem and conjunctive tissue on the inner side and phloem and parenchyma on the outer side. The resulting tissue gives the appearance of concentric rings of vascular bundles embedded in the conjunctive tissue. The anomalous secondary growth of Ducat-Boerhaavia stem showed following tissues from outside within. Epidermis. Single-layered epidermis consists of small, radially elongated cells. Multicellular epidermal hairs arise from some cells. A thick cuticle is present on the epidermis. Some stomata are also present. 2. Cortex. It is well differentiated and consists of few-layered parenchymatous hypodermis followed by chlorenchyma. Parenchyma is 3 to 4 cells deep, but generally near stomata it is only one-layered. Chlorenchyma is present inner to parenchyma in the form of 3 to 7 layers. Chlorenchymates cells are thin-walled, oval, full of chloroplasts and enclose many intercellular spaces. 3. Endodermis is clearly developed and made up of many, tubular, thick-walled cells. 4. Pericycle. Inner to the endodermis is present parenchymatous pericycle, but at some places it is represented by isolated patches of sclerenchyma. 5. Vascular bundles. Vascular bundles are present in 3 rings. In the innermost ring are present 2 large bundles. In the middle ring the number ranges from 6 to 14 while the outermost ring consists of 15 to 20 vascular bundles. Vascular bundles of innermost and middle rings are medullary bundles. All the vascular bundles are conjoint, collateral, endarc and open. 2 vascular bundles of the innermost ring are large, oval and lie opposite to each other with their xylem facing towards center and phloem outwards. Vascular bundles of inner and middle rings may show a little secondary growth. The cambium produces only a little amount of secondary xylem to the inner side and secondary phloem to the outer side. As a result the vascular bundles of these rings become slightly enlarged. Interfascicular cambium never develops between these bundles. Now here is the CS cross-section of the Boerhaavia stem and TS transverse section of the same. Hope this too helps. Anomalous secondary growth in monocot stem. A. Dracaena stem. Normally secondary growth happens in dicots and so, anomalous growth also. But, there are some monocots like Dracaena, yucca, agave in which anomalous growth is found. Normal structure. 1. Epidermal zone. It has single-layered parenchymatous epidermis which is replaced by periderm with cork cells. Cork cells are phellem or thick-walled and compactly arranged. Below this cork cambium or phelogen is found with few layers of thin-walled rectangular cells arranged in storied manner. Somewhere lenticels are also found. 2. Cortical zone. The cortex is many-layered with parenchymatous cells having sufficient intercellular spaces. 3. Ground tissue and primary stele. There is large ground tissue area made of parenchymatous tissue. Primary vascular bundles are scattered in it. Vascular bundles are oval or round leptocentric or amphivoccal and closed, in which phloem is surrounded by xylem. Anomalous secondary development. The secondary meristem or cambium originates in the deep layers of cortex or pericycle. The cambial cells are fusiform or rectangular in shape. The cambial cells go on dividing and producing secondary tissues on the inner side first, and later small amount of new tissues are cut off on the outer side as well. The tissues cut off by the cambial cells on the outer side are scanty in amount and are parenchymatous in nature. Those formed on the inner side differentiate into oval-shaped vascular bundles and radially arranged parenchyma cells. The radially arranged secondary parenchyma constitutes the conjunctive tissue. The radial arrangement of the parenchyma cells of conjunctive tissue is due to their origin by tangential divisions of the cambial cells. They may be thin-walled or thick-walled. A few derivatives of the cambium divide anticlinally, then paraclinally and even haphazardly to form xylem and phloem elements of the secondary vascular bundles. The secondary bundles are mostly amphivosal, some of them may be collateral as well. The small amount of phloem consists of short sieve tubes, companion cells and parenchyma. The xylem is made of only tracheids, usually with scalariform thickening and small amount of xylem parenchyma which have lignified walls. Here it is the CS cross-section of the stem, and now it is the TS transverse section of the stem. Hope this comments from each of our subscribers. But due to time limitations, some may face delay. And please cooperate until we can make them fine. More and more requests are on way and we are thankful on each of you for your suggestions. Hope we can make maximum of them uploaded. Microtomy essay explained. Cutting of thin sections of objects, such as tissues or cells for microscopic or ultramicroscopic examinations. Instrument used microtomes, for overcoming the limitations of freehand sectioning. Characters of microtomes. Feedback mechanism to move the material or cutting knife over a specific distance. Block holder for holding tissue block. Strong knife holder. Types. 1. Mobile knife, in which block with material remains stationary and knife move across it. Example. Bench microtome. Clinical microtome. Sliding microtome. Sledge microtome. A. Sledge microtome. Device with mobile sample holder. Shutter, which moves back and forth across a knife, with the adjustment of angle between sample and microtome knife. Pressure applied can be regulated. In modern versions for readily cutting many coarse sections, sledge is placed upon a linear bearing, used for preparation of large samples, with those embedded in paraffin for biological preparations. Typical thickness obtained, about 1/60 micrometer. For more details, the image of a sledge microtome is given here. 2. Stationary knife, in which the knife remains stationary and block moves across it. Example. Rocking microtome. Rotary microtome. A rotary microtome consists of large wheel rotation of which causes block holder to move across knife. Material mounted to block holder is fixed to rotating object clamp for orienting object to any desired position. Knife held in a rigid holder and can be adjusted through a wide range of cutting angles. Basic principles of rotary microtome. Operated by drive wheel. Knife stationary. Material, block. Holder moves across it and fixed to a rotating clamp for serial sectioning. Have specialized mechanical settings to produce sections of desired thickness. Knife uniformly sharp-edged end. Holder can be adjusted through a wide range of cutting angles. Examples: Irma-type rotary microtomes. Spencer-type rotary microtomes. Here comes another image of rotary microtome for more information.
Scattering of data displays and gives us an idea about the variation in the central value, central tendency of an individual item. Explains the disparity of data from one another, delivering a precise view of their distribution. Includes range, mean deviation, standard deviation, variance, coefficient of variation, and so on. A range, simplest of the measures of dispersion, is the difference between the highest and lowest observations in a set of data. Merits, easy to compute, simplest to understand. Demerits, X very crude measure and does not take into account all the observations. X in cases where the data is not much variable extreme values present in the data can increase the range, and in turn we gets distorted picture of variation present in the data. X not based on each and every Observations of the data. X is subject to fluctuations of considerable magnitude from sample to sample. X is not giving any idea of the inter-modal values. X cannot be computed in case of open-ended distributions. B. Mean deviation, also known as average deviation. The arithmetic mean of the absolute values of the numerical differences between the numbers of a set, such as statistical data, and their mean or median. Note mean deviation can be calculated easily by using the difference between each observations and either the mean or median obtained. Merits. Outstanding advantage: its relative simplicity, based on each and every data, and is less affected by extreme values in the observations. Comparison about formation of different distributions can be made easily since deviations are taken from a central value. Demerits. X algebraic signs are ignored, that is, method is non-algebraic. X may not give very accurate results, since mean deviation from median gives best result compared to mean deviation from mean, and median is not considered as a satisfactory measure of central tendency when degree of variability in a series is very high. C. Standard deviation is the positive square root of the arithmetic mean of the squared deviations of observations from their arithmetic mean of the whole series under consideration. Also known as root mean square deviation, and is denoted by sigma. The concept of standard deviation was introduced by Carl Pearson in about 1893. Standard deviation for ungrouped data is as follows, where n equals no of observations. Standard deviation for grouped frequency distribution is given below. Here n represents the total of the frequencies obtained. Merits. Best measure of variation and is mathematically calculated, is based on each and every observations of the distribution. Also is possible to calculate combined standard deviation for two or more groups of data. Demerits. X. It is very difficult to compute. X. It doesn't give the full range of the data to be analyzed. D. Variance is the expected value of the squared variation of a random variable from its mean value. It can also be defined as the square of standard deviation. Merits. It treats all deviations from the mean as the same regardless of their direction. Is a strong tool to locate differences between past and present performances and respond to the issue appropriately. Demerits. X. Not so easy to interpret. E. Coefficient of variation. The coefficient of variation, CV, is the ratio of the standard deviation to the mean. It is generally expressed as a percentage. Merits. Since is independent of measurement units of original data, it allows to compare two different observations. Demerits. X cannot calculate with logarithmic values. X sensitive to small mean values. Thank you all for watching.
The update of today too is a suggested video as of always. Polysyphonia. Kingdom. Plantae. Division. Rhodophyta. Class Rhodophyceae. Subclass. Floridae. Order Ceremeales. Family Rhodomelaceae. Genus Polysiphonia. Species Fastigiata. Urceolata. Platycarpa. Variegata. Elongata. General characters and structure. Exclusively marine algae. Thallus. Large. Multiaxial, perennial. Violet-red thick stuffs or dense bushes. Heterotrichous body differentiated into prostrate and aerial system. Presence of central siphon in cylindrical cells of main axis and pericentral siphons. In surrounded cells provide the name. Polysiphonia. Main axis bears two types of lateral branches. Trichoblasts, short branches. Limited growth, and long branches. Unlimited growth. Unenucleated cells with pigments are phycoerythrin. Chlorophyll A. D. Carotenoid tauroxanthin. All cells of central and pericentral siphons remain connected with each other through cytoplasmic connections, or pit connections. Reproduction. A. Vegetative. Bifragmentation. B. Asexual. Bicarpospores and tetraspores. C. Sexual reproduction life cycle. Oogamness. Most species. Heterophilic. Male. Antheridium, spermatangium. Female. Carpogonium. Antheridium. Contents of spermatangium produce a single, non-motile, spherical male gamete spermatium. Spermatia are carried by water currents to the vicinity of female sex organ. Carpogonium. Terminal cell of carpogonial filament. Differentiates to form carpogonium. Procarp developing carpogonium. Ready for fertilization. Fertilization. Male gametes from antheridium reaches basal swollen portion of carpogonium and fuses with egg to form diploid nucleus. Represents beginning of diploid phase or formation of carposporophyte. Post-fertilization events. Carpospores from carposporophyte germinate into diploid, tetrasporophytic plants. Tetrasporophytes bears tetrasporangia, from which tetraspores are formed. Note tetraspores are haploid spore, formed by reduction division. Tetraspores on germination produce male and female gametophytic plants. Thus life cycle is completed. Here comes the diagrammatic representation of the life cycle. Thank you all.
Custard apple family. This group of plants is home to some of the most delicious and exotic fruits you've ever seen, like the sugar apple and the soursop. But Anonaceae is more than just tasty treats. Its members have been used for centuries in traditional medicine, and have even been found to have potential anti-cancer properties. Join me as we delve into the world of Anonaceae, and discover the secrets of these incredible plants. Family, Anonaceae. Class, Dicotyledons. Subclass, Polypetalae. Series, Thalamiflorae. Order, Ranels. About 108 genera. 2,400 species. Distribution, mostly tropical. Habitat, mesophytic. Habit, shrubs, trees. Examples, Tilda trees, Polyalthea. Tilda shrub, Tilda hook climber, Artabatres, Uvaria. Tilda monopodial growth, Polyalthea. Tilda sympodial, Atrabatricin. Aromatic wood due to oil passages in stem in the parenchyma cells. Leaves, simple, alternate exstipulate, petiolate with entire margin. Inflorescence, solitary, fascic lead and others. Flower, greenish yellow-brown, pedicellate, bisexual, actinomorphic, dichlamydous, hemicyclic, hypogynous. And trimerous, calyx and corolla distinguished by size only. Hence is called perianth. Tilda sepaloids 3. Small, polysepalis with valvate. Estivation, triangular in shape. Tilda petaloid. Petals 3 or 6, polypetalous with valvate. Estivation, imbricate in some, may be thick and fleshy. Shape of thalamus varies in Anonaceae. On the prolonged thalamus, stamens and 7 pistils are arranged spirally. Androecium. Stamens numerous, free, spirally arranged, closely packed at basal part of thalamus. Tilda filament. Short and thick, with adnate bithyx anther. Tilda dehiscence longitudinal and extrorse. Tilda connective is prolonged above the anthers to form an elongation called hood, so, hooded stamens. Genetium. Ovary superior, multicarpillary. Apicarpus, spirally arranged on thalamus above the stamens. Tilda pistil with individual ovary, style, stigma. Inconspicuous. Fruit. Aggregate fruit, eterio of berries. Seed. Endospermous, hard testa. Tegmen produced to numerous infoldings into the endosperm, ruminate endosperm. Embryo, small, economic importance, edible, have medicinal value, such as ABTI bacterial, anti-cancer properties. Have ornamental value, wood or timber used for housing and carpentry. Habit, a small tree. Root, tap root, branched. Stem, aerial, woody, branched. Leaf, simple, alternate, exstipulate, entire, oblanceolate, obtuse. Inflorescence, axillary, 2 or more flowers arise in the axil of each leaf. Calyx, sepals 3, polysepalous, united at the base, valvate. Corolla, petals 3, polypetalous, fleshy, clay yellow, valvate. Androecium, stamens numerous, spirally arranged on a conical receptacle, filaments short, anthers. Oblong, adnate and appendaged. Genetium, multicarpellary, apocarpous, spirally arranged, superior, each unilocular, ovule 1, basal placentation, style short, stigma long, tapering, papiose. Fruit, an aterio of berries. Floral formula and diagram are as follows. Example, Cananga odorata, habit, tree, stem, erect, woody, aromatic, cylindrical, bark is smooth. Leaves, simple, alternate, exstipulate, aromatic pedicellate, base is often unequal, waxy margins. Inflorescence, raceme, 2 to 6 flowers on short, leafless, axillary shoots. Flowers, ebractiate, pedicellate, regular, actinomorphic, bisexual, hypogynous, spirocyclic. Calyx, sepals 3, valvate. Fruit, pendulous, consisting of many, 7 to 16, separate, globose obovoid. Monocarps. Seeds. Scale, can be defined as the geological time scale is a framework used by geologists and paleontologists to divide Earth's history into distinct time intervals based on significant geological events and changes in life forms. It is organized hierarchically, with larger units subdivided into smaller units. Eon, era, period, epoch. Here is the image showing the geological time scale with different era, periods and epochs in detail. The history of Earth is divided into several eras, including the Precambrian, Paleozoic, Mesozoic, and Cenozoic eras. The geological time scale provides a way to date rocks and fossils, allowing scientists to determine the relative ages of geological formations and the fossils found within them. The divisions in the time scale are often associated with significant geological events, such as mass extinctions, changes in climate, and the shifting of continents. Each era is characterized by the types of life forms that existed during that time, making it a valuable tool for understanding the history of life on Earth. The current era, the Cenozoic, is the era in which we live and is marked by the dominance of mammals and the appearance of humans. Geologists and paleontologists use the geological time scale to study Earth's history, identify geological strata, and correlate fossils with specific time periods. It provides insights into the evolution of life on Earth and helps scientists understand how species and ecosystems have changed over millions of years. Understanding the geological time scale is crucial for studying Earth's history, interpreting geological formations, and dating fossils accurately in the field of paleobotany. Fossil formation. Fossils are the preserved remains or traces of ancient organisms, providing valuable insights into Earth's history and the evolution of life. A fossil can be an entire organism, a hard part of it, a mold or cast of an organism or its parts, or even the imprint of a leaf on stone. The process of preservation of dead organisms or their parts in the form of fossil is known as fossilization. The totality of fossils, both discovered and undiscovered, and their placement in fossiliferous rocks with fossil formations and sedimentary layer is known as fossil record. Fossilization process is effected by a number of factors, such as 1. The nature of the plant, whether it is thin, delicate, or hard. 2. The rapidity with which the plant material is prevented from decay by organisms. 3. The changes to which rocks are subjected during or after the burial of the plant parts. 4. The extent of mineralogical importance. 1. Role in soil conservation. Some like mosses checks soil erosion. Grows luxuriantly and forms thick mats on soil surface. The tangled stems and rhizoids of these help to bind particles tightly up to a depth of about 15 to 20 centimeters, which prevents the sheet erosion even on steep hillsides. Moss mats also reduces the impact of rainfall and holds much of the rainwater. 2. Role in soil formation and growth of vegetation.
lichens and mosses grows in the cracks and crevices of rocks causes the weathering of rocks with the help of the acids they release. The death and decay of these also contribute to fertile soil formation. 3. Role in rock succession. Liverworts, mosses, lichens serves as pioneers in lithosphere. On rock, they initiate soil formation in bare rocks where other plants colonize later. 4. Role in bog succession. Sphagnum and other hydrophytes, established in water bodies, transform water bodies into quacking bogs which later on develops to swamp forests due to the colonization of large trees. Economic importance. 5. Role in monitoring and controlling pollution. Some mosses, sphagnum, both alive and sun-dried, can absorb metals. Thus the heavy metal accumulation enables them to be used as indicators of aquatic, atmospheric pollution as well in mineralogical surveys. Can be used as indicators of air pollution since are sensitive to air pollution. Their sensitivity increases from terricolous to corticolous species, are also sensitive to water pollution, hence can act as an indicator of water pollution too. Some pollution tolerant aquatic species, such as Amblystygium riparium, Pontinellus antipyretica, Riccia, Frosti, Funaria hygrometrica, are recommended to monitor the level of water pollution. 6. Role as sources of antibiotics. Aqueous extracts of Conchocephalus coricum antibiotically active, proposed by Hayes 1947. Atrichum, polytrichum, sphagnum have antibacterial activity. About 52 species of tested bryophytes such as Anemodon, Rostratus, Orthotrichum rupstrae, and so on have antibiotic activity against 12 species of microorganisms. 6. Role as medicine. Marchantia polymorpha. 4. Treatment of pulmonary tuberculosis and liver diseases. Decoction prepared by boiling dried sphagnum for treatment of acute hemorrhage and eye diseases. Tea made of polytrichum commune to dissolve kidney stones. Peat to treat skin diseases. Many are used as antibiotics as detailed above. Some mosses, sphagnum, can be used as absorbent bandages in hospitals in place of cotton. 7. Role in horticulture. Peat is added to clayey soil to improve its texture. It keeps the soil porous and prevents its caking. Since peat is one of the important sources of humus used to increase fertility, it also improves the water holding capacity of the soil. Sphagernum, dry, live, and peat are extensively used as substratum for seed germination, a packing for grafting scions to protect them from drying, packing material for shipment of live plants and other plant parts, since they retain moisture for long time and afford protection from heat and cold. 8. Role in experimental botany. Important research tool in fields of genetics, morphology, physiology and so on. The mechanism of sex determination in plants was discovered for the first time in liverwort, Sphaerocarpos. 9. Role as packing material. Many dried mosses, sphagnum, used as fairly good packing materials for packing fragile articles, such as glasswares, seedlings, plant cuttings, etc. Note: the compacted, partially decomposed and carbonized deposits of bryophytes are called peats. 10. Importance of peat. Used as fuels. Cellulose present in peat is broken down into sugar which is fermented to alcohol. Can be used for the manufacture of paper, fabrics, artificial wood. Sphagnum along with peat have a great role in horticulture. Used for production of acetic acid, paraffin, carbonic acid, etc. Note: please follow the rules when only be a starter for you dears, and it includes the notes on evolutionary significance of Peripatus. Evolutionary significance of Peripatus. Phylum Onychophora, in which Peripatus included, is a monophyletic taxon derived from a single ancestral line. Common ancestry shown by Common possession of mandibles on second head segment. Oral papillae. With slime glands on third segment and widely spaced ventral nerve cords. Connected by numerous commissures and tufted tracheae. They, onychophorans, probably arose from Cambrian panarthropod ancestors as an exclusively terrestrial line. Shows morphological similarities that suggest relationships with both annelids and arthropods, but more closely related to arthropods. Peripatus, a living fossil because living members of this animal group appears similar to fossil species that lived 570 million years ago. Also as a connecting link between Annelida and Arthropoda. Features shared with arthropods includes presence of antennae, salivary glands, absence of internal segmental septa and so on. Features shared with annelids includes worm-like body, simple eyes, absence of distinct head and so on. Characters similar to both includes segmented body, paired appendages, ladder-like nervous system and so on. Dendrifying, nitrogen fixing, cellulose splitting and sulfur bacteria, actinomycetes, fungi, algae, protozoans, nematodes and so on. Bacteria, major group which includes autotrophs and heterotrophs. Example, Micrococcus, Bacillus, Pseudomonas, Clostridium, Arthrobacter, Achromobacter, Flavobacterium, Mycobacterium and S0 on. Actinomycetes abundant in soil rich with decomposed organic matter. Example, Streptomyces, Nocardia, Micromonospora and so on. Fungi abundant in moist soil and helps in improvement of nutrients in alkaline and neutral soil. Example, Aspergillus, Alternaria, Botrytis, Cephalosporum, Cotomium, Rhizopus and so on. Algae helps in nitrogen fixation, soil conservation, improvement of soil structure and so on. Example, Chlorella, Chlorococcus, Anabena, Nostoc and so on. Protists, numerous in topsoil and bacteria-rich soils. Example, Alluation, Bodo, Circomonas, Heteromeda, Nuclearia, Oxytricha, Vorticella, Pleurotricha and so on. Rhizosphere, area of soil immediately surrounding root systems, modified by growth, respiration, nutrient exchange and so on by plants and other microbes. Rhizoplane or root surface. Further, zone, effect and influence of root system on microbial population determines the number of them in rhizosphere. Soil microbes helps in promoting branching, spreading of roots and formation of root hairs. Rhizosphere environmental issues, which includes not only study of physical and biological features of the environment, but also the aspects of the issues related to the social, cultural, political and economic aspects of the environment. Scope and relevance enables us to understand our environment. It provides a key to know how to manage the living world in a better way. Creates awareness about harmonious relation between man and environment. Advocates protection of nature from man for man through intelligent monitoring and management of the interrelation between man and nature. Need for environmental awareness. In this scenario of increasing anthropogenic destructions, environmental protection and awareness programs are very much in need. The critical situation is that while awareness and analysis are improving, appropriate actions are becoming more and more conspicuous by their absence or inadequate. The notes are based in the topic Cell Biology Semester 5 Core Botany syllabus. Botany complementary students too can also use this. Cell biology, the branch of science that deals with the morphological, physiological, biochemical, genetical and developmental aspects of cells. History, invention of microscope, Janssen and Hans. Cell discovery, Robert Hooke, 1665. Discovered sperm cells, RBCs and several kinds of microorganisms using a single, lensed microscope. Cell theory, Schleiden and Schwann. Described somatic cell division and applied term mitosis 1882. Advanced the germ plasm theory, Weissman. Chromosome theory of heredity, Sutton and Boveri. Invention of electron microscope, Knoll and Ruska 1928. Invention of phase contrast microscope, Zernick 1940. Note: the key events in the milestones in the history of cell biology are the cell theory proposed by Schleiden and Schwann, which provides the concept that living organisms are formed of cells and cell products, and the protoplasm theory introduced by Purkinje. Properly store, search and retrieve any type of data. Share large amounts of data and support large-scale analysis by easy access and will data updation. Biological database carries information such as DNA, RNA, protein sequences, 3D images, Organism, specific information and information from literature. Classified into primary, secondary, derivative, nucleic acid database. Organize and distribute nucleotide sequences from all available sources. Example: EMBL, GenBank, DDBJ, European Molecular Biology Laboratory, EMBL. European Node of 3 Ways International Nucleotide Sequence and Annotation with comprehensive global coverage. Key goal is to integrate nucleotide sequence and annotation into wealth of bioinformatics resources also offered at the EMBL and elsewhere. Access points to EMBL nucleotide sequence database data include SRS. Homology search tools. The Sequence Version Archive, SVA. FTP site. The Genome's web server, for completed genomes, and sequence retrieval by accession number, DB Fetch. WSDB Fetch and NetServe. It offers a wide range of opportunities to search and download data through the help of access points. GenBank. Most comprehensive and annotated collection of all publicly available DNA sequences. DDBJ. EMBL and NCBI exchange data on daily basis. Important source of data for GenBank. Direct submission from scientists, and the options for submitting data to GenBank include Bankelt. Sequin. Macrosend. TBL2ASN. Keywords used in GenBank files includes locus, definition, accession number, version, keyword, source, organism, reference, authors, journal, PubMed identifier references, features and so on. DDBJ. DNA Data Bank of Japan was established at NIG National Institute of Genetics. Japan. Works in collaboration with EMBL and GenBank and share data and information on Internet. Principal purpose: to improve quality of INSD, International Nucleotide Sequence Database. As public domains. Open to public and commonly shared through worldwide. Collect DNA sequence data mainly from Japanese researchers. Sequence submission tool. Sakura. Protein sequence database. Collection of sequences including translations from annotated coding regions in GenBank, RefSeq and TPA, as well as records from Swissprot. PIR, PRF and PDB. Contains information derived from the primary sequence databases. Example: PIR, Swissprot, Uniprot, PIR, Protein Information Resource, integrated public bioinformatics resource to support genomic and proteomic research and scientific studies. Established by NBRF as a resource to assist researchers and customers in identification and interpretation of protein sequence information. PIR offers a variety of resources mainly oriented to assist the propagation and standardization of protein annotation. Swissprot. High-quality annotated repository. Created by Department of Medical Biochemistry, University of Geneva and developed and maintained by European Molecular Biology Laboratory and Swiss Institute of Bioinformatics, SIB. It cross-reference with several other databases including nucleic acid and protein structure databases. Classifies data into 2 classes, core data and annotation. Uniprot. Mission: to provide scientific community with a comprehensive, high-quality and free accessible resource of protein sequence and functional information in which many entries are derived from genome sequencing projects. It comprise EBI, European Bioinformatics Institute, SIB, Swiss Institute of Bioinformatics and PIR Protein Information Resource. Comprises of 4 components, each optimized for more different uses. They are: Uniprot Knowledge Base, Uniprot Reference Clusters, Uniprot Archive, Uniprot Metagenomic and Environmental Sequences, Uniprot Knowledge Base Central Access Point for extensive curated protein information, including fusion, classification and cross-reference. Uniprot Reference Clusters databases provide clustered sets of sequences from the UniprotKB and selected Uniprot archive records to obtain complete coverage of sequence space at several resolutions while hiding redundant sequences. Uniprot Archive Comprehensive Repository. Used to keep track of sequences and their identifiers. Uniprot Metagenomic and Environmental Sequences was a repository for metagenomic and environmental data. Protein Data Bank, PDB. Repository of information about the 3D structures of large biological molecules, including proteins and nucleic acids, data derived from X-ray diffraction and NMR studies. Established in 1971 at Brookhaven National Laboratory when RCSB, Research Collaboratory for Structural Bioinformatics became responsible for the management of PDB, it came to have an international community of users including biologists, other scientists, students, educators, media writers, illustrators, textbook authors and general public. Funding members: RCSB PDB, USA, PDBE, Europe, and PDBJ, Japan. Keywords in PDB file includes header, title, compound, source, author, Revdar, Sprayed, JRNL, Secras, Atom, Heditum and so on. Model Organism Database MOD, dedicated to the provision of in-depth biological data for intensively studied model organisms. Allow researchers to easily find background information on large set of genes. Plan experiments efficiently. Combine their data with existing knowledge and construct innovative hypotheses. Allow users to analyze results and interpret datasets, and the data they generate are increasingly used to describe less well-studied species. Share common approaches to collect and represent biological information wherever possible. Mods are often required for customize, capture, display and provision data, generate, source and collate species, specific information integrating expert knowledge with bioinformatics. Biodiversity Database, Species 2000. Federation of database organizations working closely with users, taxonomists and sponsoring agencies. Established by International Union of Biological Sciences, IUBS. Goal to provide a uniform and validated quality index of names of all known species for use as a practical tool. The index are used to provide an electronic baseline species list for use in inventing projects worldwide. 2. For an Internet gateway to species databases worldwide. 3. A reference system for comparison between inventories. 4. A comprehensive worldwide catalog for checking the status, classification and naming of species.